Thank you again, everyone, for joining our March Pepper Center rounds. We're thrilled to have Dr. Doug Vaughan here to speak with us today. Dr. Vaughan is the Irving Cutter Professor of Medicine Emeritus, Director of the Potocsnak Longevity Institute at Northwestern University Feinberg School of Medicine, and Chief Academic Officer at Northwestern Medicine. From 2008 to 2023, he served as Chair of the Department of Medicine at Feinberg. During that time, he also served as the Physician-in-Chief of Northwestern Memorial Hospital. His research program includes basic translational and clinical research focused on the role of the plasminogen activator system in cardiovascular disease and aging. Vaughan's research team identified the first autosomal dominant gene mutation that protects against biological aging in humans. This loss of function mutation in SERPINE1 provides the first evidence that reduction in a component of the senescence messaging system impacts key molecular and physiological aspects of aging and the lifespan. His group is now focused on the development and testing of PI1 inhibitors, small molecules, biologics, and siRNA that protect against the development of senescence and age-related pathology. These agents have the potential to delay or reverse aging-related morbidities in humans. With that, Dr. Vaughan, I'll let you take it away.
Thanks very much, Sarah. And thank you and thank Mike Wolf for inviting me to speak to the group today. Can you hear me all right? Great to be here today. It is the first day of spring. So for those of you that haven't been in Chicago, our city is really on a grid. The streets run north, south, east, west, and here for a couple days we have something called Chicago Hinge. So at sunrise and at sunset, you can see the sun quite fully aligned with the avenues that run east, west in Chicago. It's quite a sight, and maybe thousands of years from now, humans will write about the strange, mysterious alignment of avenues in this large metropolitan area. So we're circling the sun, and every time we complete an orbit, we add a year to our chronological age. Actually, I completed an orbit yesterday. But this is an important point. We think that if you think about aging and you think about how it happens to all of us, we have to think about the fact that there is a difference between chronological and biological age. And I'm going to level set the conversation at the beginning here today to make sure that we all can see so that you all can see what I'm talking about here. So your chronological age is something we're all familiar with. Every time you have a birthday, you add another year to your life. There's nothing you can do about it. It happens. It's relentless. It's steady. I call it cosmically indifferent. You're going to get older as long as you're alive in terms of chronological years. But is that the whole story? I think we've learned now, and we're learning more every day, that it is not the whole story, that there is a biological age as well. It's the clock within us. It really registers changes in our function and our systems as over time, but it doesn't necessarily do it with the same kind of periodicity and rigor and pattern that chronological age occurs. It occurs in a different kind of way. It doesn't count years, it counts change. So this slide sort of is a simple representation of the fact that people experience biological aging at different rates. And you're all familiar with this fact. You know people in your life, you know people that you work with, you know people in your family that fall on one or the other trajectories that I've illustrated here. Most of us have our chronological age and biological age line up pretty well. There's not much difference between the two. But there are certainly people that appear to be superagers. We have a big program around superagers here at Northwestern through the Musulam Center. There are people that retain their functionality well into their 90s. We're still learning the mysteries of that and why that happens. There are a lot of things that appear that play a role in that. On the other hand, on the other side of the spectrum are people with accelerated aging, people that show an enhanced rate of biological aging, maybe due to chronic conditions or circumstances like HIV or chronic kidney disease, Maybe it's where they live. Maybe it's what they're exposed to. Maybe it's what they eat and drink, all kinds of things. So different trajectories, different rates. And the fact that there are different rates, I think tells us in some way that aging is malleable in some way. It doesn't have to be relentlessly aligned with your chronological age. Other things are happening in the world right now and over the last several years. This is a photograph I took a couple years ago, flying back from London over the southern tip of Greenland. Now, I didn't know Greenland would be so interesting at the time when I took this photograph a couple years ago, but that's a topic for another day. But as I looked out the window, I snapped a photograph of two gigantic glaciers that were merging and going out to sea. And this concept of convergence is something I want to spend a moment with. I think we're at a unique moment in human history. On one hand, the biology of aging has been largely demystified. It's not complete, but we know a lot about what drives aging and the biology of that process. Simultaneously, we've learned how to measure biological age in people with a precision that never existed before. And when those two things come together, it makes it possible to start thinking about and imagining that we can actually bend the curve of aging in our species. And this is the moment we're in 2026. We're thinking about it. People all over the world are thinking about it. There's an enormous amount of money being invested in figuring out how to slow down aging in people. Some of the largest corporations on the planet are spending hundreds of millions, even billions of dollars to get into this game. And there's no doubt about it. In the next few years, we're going to find ways to slow down aging in human beings. Why is it important? I think it's really relevant for the Pepper Center organization and all of us in medicine because the idea is simply illustrated right here. And I think you've probably seen a version of this slide somewhere before. The geroscience hypothesis is largely funneled around the concept that aging is a malleable process. Age is the most important risk factor for almost every disease we deal with in adult human beings, whether it's cardiovascular disease. I'm a cardiologist, but it's true for cancer. It's true for metabolic disease. It's true for neurodegenerative disease. There's a very common denominator that age is the most important risk factor. And it's not just chronological age, it's biological age. And we can't do anything about chronological age, but if we can do something about biological age, if we can slow that down a little bit, we might be able to actually extend the health span in people and provide more disease-free years. It's important to say that because while chronological age is a great component and determinant of overall risk, the measures of biological age that have come out over the last few years are even a better predictor of age-related morbidities. So that plays into the idea that biological age is actually the driver of age-related morbidity, and can we do something about it? That's the big question, and that's one of the questions we're trying to help answer here in our Longevity Institute. I want to spend about 10 minutes with you today telling you about how I got into the field of aging, how I found it, maybe how it found me, and tell you a story that I think helps inform, you know, the perspective that I use in coming at this challenge and this question and this phenomenon, and part of what helped inform what we're doing in our Longevity Institute here. So turn the clock back a few decades now. I was completing my cardiology fellowship in Boston. It was a really exciting time in cardiovascular medicine. A drug called TPA had arrived on the scene. Tissue plasminogen activator was developed and marketed by Genentech. It was one of the first big recombinant drugs. And what did it do? It helped dissolve clots rapidly, efficiently by harnessing the plasminogen activator and clot dissolving system of our body. I was fascinated by this phenomenon. I was fascinated by the plasminogen activator system and how nature made a clot-specific, clot-dissolving system. So the person that gets, I think, most of the credit for discovering TPA and pushing its development in the clinic was a physician scientist at the University of Leuven in Belgium named Desiree Collin. So after I finished my cardiology training, I actually got a grant from the American Heart Association, and I went to Belgium to work in his lab. I wanted to be one of the cardiologists that was an expert in plasminogen activation and the use of these thrombolytic agents. I actually was the fellow on the first study in human beings testing TPA in people with acute pulmonary embolism. So there I went over to, to Leuven in late 88. The lower panel on the right shows a picture of the laboratory. It was a big group. Here's Desiree Collin right here. Here's a young version of me standing next to him. I don't know why I got to stand next to him. I think everybody in the lab was kind of frightened of him, so nobody wanted to stand near him. So I was the one that ended up standing there. While I was there, I went with a project in mind, but I was standing in the hallway with Desiree early on in my tenure there, and he asked me what I wanted to do, and I gave him an answer that, frankly, I don't think he was too impressed with. And he said he looked down the hall and he pointed to a bank of freezers, and he said, "You know what? A few years, recently, this inhibitor of TPA has been identified called Pi1, plasminogen activator inhibitor type 1. He said we have several milligrams of it in that freezer over there." He goes, "Nobody knows what it does. Nobody knows its actual biology other than the fact that it can inhibit TPA. It might have something interesting about it." He said, "Why don't you work on that?" And it took me about 5 seconds, and I sort of did a Pete Davidson thing and said, "Okay, I'll do it." And almost 40 years later, I'm still working on it. And it was just that chance opportunity to meet with Desiree and to have an opportunity to work and study the biology and function of the protein that you see down in the lower right-hand panel here. That's a ribbon image of plasminogen activator inhibitor. It's a member of the serpin superfamily of serine protease inhibitors. It's made in your liver, it's made in your fat, it circulates in your blood in really low concentrations. It's got a short half-life in plasma, and it's cleared in the liver. And you can see it's a beautiful protein. You can see why I still dream about it at night. So very early on, I adopted Pi1 as the focus of my research program and the independent program that I started to build when I got back to Boston and when I moved to Vanderbilt in 1993. But in late 1992, a very interesting short article was published in the New England Journal of Medicine, describing a young woman, a young girl really, in an Amish community in northeastern Indiana that had an unusual bleeding disorder. And it turns out she had a complete deficiency of Pi1. Now, she didn't bleed spontaneously. She's not a hemophiliac, but she exhibited the physiology of a clot dissolving system that was a little bit uninhibited. So she didn't bleed spontaneously, but if she was injured, her primary hemostasis was fine. She could form a clot. But a day or two later, that clot would get broken down because her clot dissolving system was a little bit too efficient. And it turns out that was the physiology of that. That was physiology manifested by having a complete Pi1 deficiency. It turned out she was a member of a larger kindred with other family members that had complete or partial Pi1 deficiency. Now, when I read that paper, I made a note to myself. I said, someday I want to go study those people. But I didn't know what the question was I wanted to ask. I knew that would be interesting because I had already declared to myself, at least personally, that I was going to work on Pi1 in my career. Why wouldn't you want to go work with the population of individuals, human beings that have Pi1 deficiency? So I filed it away, and we'll talk about it some more in a minute. Not much longer. A few years after that, as we were churning along and doing all kinds of things, looking at the functional regulation of Pi1 in cells and in tissues, a really interesting article was published in Nature describing a novel new mouse model that exhibited a progeroid sort of phenotype. And the mouse was called Clotho. These investigators in Japan had inadvertently altered the structure of a previously unknown gene. And by doing so, they generated and engineered a mouse with a very novel phenotype. Short lifespan, growth retardation, malignant emphysema, infertility, hypoactivity, vascular calcification, renal sclerosis, all kinds of manifestations that looked like aging. One of the papers that they published included kind of offhand a report, some evidence that actually that pi1, that gene, that protein that I was interested in, was increased in tissues, both in tissues and in blood in Clotho deficient animals. Around the same time, the first publications were coming out indicating that pi1 was actually a key marker of senescence in cells, in endothelial cells and other cells in the body. It was one of the first representations of the of SASP, so the linkage between pi1 and senescence and pi1 and aging-related phenotypes registered with me. How do we? So we came up with an idea how to test it, and I actually asked the question, and we asked the question together: does pi1 contribute to the aging phenotype in Clotho? Simple question, hard to answer. We took several years for us of breeding pi1 deficient and Clotho deficient animals to try to sort out the role of pi1 in the aging-like phenotype in mice. And we ended up publishing this in PNAS in 2014. Here's the key finding. There are others. By reducing pi1 in Clotho deficient animals, we protected them from many aspects of the aging-like phenotype. But look at this. This is their survival. So the green line here represents the demise of Clotho hypomorphic animals. They're all dead by just over 100 days. Wild type mice go on for quite some time. They live more than 2 years, and that's the line on the top. But look what happened when we took pi1 out of the equation. Either completely homozygous pi1 deficient mice or partial deficiency of pi1. The impact was pretty similar. Overall, we found that there was nearly a quadrupling of their lifespan by taking pi1 out of the equation. And the amazing thing at least to my mind, was that pi1 deficiency was enough to have an impact on the aging-like phenotype. So this was a eureka moment for me. This is almost a selfie of me as this idea came to mind after seeing the data of the role pi1 played in aging in Clotho. Probably maybe I think of myself and I think of my career. I think this is maybe the most important aha moment that I've experienced, because it took me back to that paper that I had filed away a few years earlier. That report that there was a community in the United States, not far from Chicago, that actually harbored a null mutation in the gene that codes for pi1. So over in northeastern Indiana, in Adams County, in and around Bern, there is a community of old order Amish that immigrated there from the area around Bern, Switzerland, in the middle part of the 19th century. They're quite traditional in their culture. No combustion engines, no electricity. They get around by horse and buggy. They speak a Swiss-German dialect with each other. Um, it's a founder population, and I'm going to talk about that some more in a minute. So what you see on the lower left is the original description of the pedigree in the New England Journal paper published in 1992. The proband is highlighted with an arrow, but you can see there are lots of individuals in that kindred that have partial or complete pi1 deficiency. The deficiency of pi1 is due to a simple typographical error in their DNA replication. There's a dinucleotide insert. There's an extra adenine here and an extra thymidine here. Bring a simple dinucleotide insert into the coding sequence of pi1. That puts the coding off kilter and generates a premature stop codon. So if you have this genetic insert, then that particular copy of your pi1 gene is rendered inactive. If you have two versions of that, you're homozygous. If you're one copy of that, you're heterozygous for this abnormality. This is really remarkable. I'd like to spend just a minute talking about this. So if you go out in the world at large and try to find human beings with a loss of function variant in SERPIN E1, the gene that codes for pi1, you have to sort through a lot of people. It's maybe 1 in 100,000. It might be even less than that. But if you go to Adams County, Indiana, 10% of the Amish in that community carry this loss of function variant. It's enriched 10,000-fold over the world at large. There's nothing like it in the world. All the individuals that carry the genetic variant can trace their ancestry back to one guy. In the middle part of the 18th century, he married into the community. He had 18 children with 2 wives. The wives were sequential, not simultaneous. So his first wife delivered 9 children, the second one 8. Uh, and he had 142 grandchildren. And you fast forward through the generations since then, you can calculate that there are uh, certainly thousands of individuals that are walking around with pi1 deficiency as heterozygotes, but it's completely unknown to them. They do not have a bleeding phenotype. They don't have any outward issues related to having that genetic deficiency. So after we did the Clotho paper, we were able to get a grant from the Heart Lung Blood Institute to actually advance and do a large phenotyping study in members of the community, together with Amy Shapiro and Sweta Gupta from the Hemophilia Foundation in Indiana, who had been caring for other members of that Amish community with other hematological disorders over many years. And in early May of 2015, we took a large group of people. Most of them are in this photograph, almost 40 members of the team, and we set up a complicated, complex laboratory in a community center where we did all kinds of things, measured a variety of different functional and physiological measures, took blood samples, did all kinds of things to ask the question, does pi1 deficiency protect against aging? So in 177 people, we actually had 43 heterozygotes in that group. We had a few homozygotes as well. And actually, on that day in early May of 2015, I sat down with the original proband and had a cup of coffee with her. And I looked at her and I told her, I've been thinking about you for a long time. And she was kind of amused and maybe a little worried, thinking, oh, that was kind of a strange thing to say to her. But she said, I don't want to be famous. I just want to have children. And she was in her 30s by then. So, but it was a remarkable moment in my life, and it registered in a powerful way that, I can only tell you how fortunate I feel to be able to actually study the impact of genetic deficiency in the gene that I'm interested in, the gene that I'm fascinated by. And the community only lives 3.5 hours from Chicago. Now, if they were in Timbuktu or somewhere in Kyrgyzstan or who knows what, we couldn't have done this, but we could drive over there and actually study the effects of lifelong pi1 deficiency in human beings. And Mother Nature is doing a remarkably interesting natural experiment over there. So this kind of summarizes what we found with regard to pi1 deficiency and how it impacts upon aging and aging-related morbidity. This was published in Science Advances now in 2017. Pi1, null mutation in SERPIN1 protects against biological aging in humans. And what's that benefit look like? Well, Our primary endpoint in the study was telomere length. That was the gold standard for measuring biological age in 2015. And the telomeres of the carriers were 10% longer than the unaffected kindred. They also had remarkable evidence of epigenetic preservation. They had lower fasting insulin levels, significantly different. And many of you, I'm sure, are aware of the fact that insulin and insulin related factors are key drivers of aging in all kinds of species, from worms to mice. The Amish carriers have lower fasting insulin levels for their lifetime. No surprise that they're protected from aging in some way. More recently, we found things like they have lower hemoglobin A1C. They're largely protected from diabetes, uh, almost a 90% reduction in diabetes in the carriers compared to controls in Adams County. They have preserved cardiovascular function in a variety of different ways. I'll talk about that in a minute. They actually have lower PCSK9 levels and they have a longer lifespan. They live about 10 years longer than their unaffected kindred. So conceptually, what we think Pi1 does and how this works out is in this figure in the middle of the slide, that there's an optimal level of Pi1 in all of us. And if you go beyond the upper and lower limits of that optimal zone, there are maladaptive effects. If you have no Pi1, you have the bleeding disorder that I talked about a little bit ago with the original seen in the original proband. But if you also have no Pi1, you get cardiac fibrosis, whether you're a human or a mouse. And we've published that work, and we've actually brought several members of the community over here that have complete Pi1 deficiency and they have cardiac fibrosis at a young age. They're also, Pi1 is a key component of your innate immune system. It blocks the invasiveness of influenza and COVID. So they are more likely to be susceptible to viruses. On the other hand, on the other side of the screen are all the aging related morbidities that Pi1 contributes to in a major way, including cardiovascular aging, clotting. We've engineered mice that overexpress a stable form of human Pi1, and they spontaneously clot their coronary arteries. Metabolic disorders are not only represented by and correlate with Pi1. Pi1 is a driver of metabolic disorders, and I actually don't think you can have the metabolic syndrome without a high Pi1 level. Pi1 contributes to emphysema and interstitial lung disease. Pi1 is also an important contributor to cognitive decline and probably Alzheimer's disease. And the problem here in the 21st century is there's a lot more Pi1 walking around on the street than there was 50 years ago or 100 years ago. Well, what's that mean? The key physiological determinant of your plasma Pi1 level is your body mass index. And the higher your body mass index, the higher your Pi1 levels. And the more you have Pi1 around, you're going to be predisposed to these aging related disorders. So Mother Nature engineered us to have very low Pi1 levels. She wasn't thinking about a world where, half of the population is obese. But here we are in the 21st century, and a component, a factor, a protein like Pi1 plays into 21st century morbidities in a way that was probably unimaginable before. I want to spend just a minute for you to help you understand how Pi1 gets involved in all these different aspects of aging and morbidity, because this is an evolving story, but it's a very, very important component of it. Pi1 regulates the proteolytic processing of all different kinds of peptides in our body. And we've made some of the discoveries that are listed on the slides have come some have come from other laboratories, but this makes Pi1 a proteolytic rheostat of aging. Because why? Well, Pi1 regulates the proteolytic degradation of factors like IGF-BP3. This is a key factor that drives senescence. If you don't degrade your IGF-BP3, you got a greater stimulus for senescence in cells throughout your body. So Pi1 augments that senescent stimuli. And there's another one, BDNF. BDNF is in our brain. It comes out in a proform. It has to be activated to the active form of the peptide by TPA. TPA in your brain. Pi1 blocks that proteolytic processing. Neuroplasticity is regulated by Pi1, and memory is regulated by Pi1. HGF, a very important factor in cellular proliferation in the liver and other organs, HGF has to be processed by UPA to generate its effects by binding to its obligate receptor. Pi1 blocks that function. We found out by studying the Amish population that Pi1 plays a role in the proteolytic degradation and clearance of PCSK9. There are companies that are being developed suggesting that we should all have our PCSK9 CRISPRed early on in life to protect us from cardiovascular disease. Well, here, Pi1 deficiency not only lowers PCSK9, it protects you from diabetes. I would argue that Pi1's a better target for CRISPR, but for another time. Pi1's involved in lipid metabolism in other ways. So these are pretty exciting, pretty interesting, and it puts Pi1 as a lever, really a node that regulates all kinds of pathways and functions that are important part of aging. I'll just spend a moment on this slide because this really speaks to some of the fundamental molecular mechanisms that contribute to the role of Pi1 in aging. So Pi1 has cell autonomous and non-cell autonomous effects on senescence, for example. Just recently, it's been published that Pi1 blocks the linkage of p53 to ubiquitin inside cells and protects its degradation. So Pi1 augments p53 dependent senescence. And actually, Pi1 is a downstream target of p53, so you get to have a feedback loop. p53 drives Pi1, Pi1 feeds back and protects the degradation, prevents the degradation of p53. So you get an amplifying circuit. Pi1 impairs the nuclear retention of cyclin D1 and alters cell cycling. Pi1, I already mentioned, has an effect on IGF-BP3 stabilization and clearance, and that's a non-cell autonomous effect. The HD effect I just mentioned, there's a really interesting story developing now that Pi1 promotes the expression of PDL1 in cells, and in particular in senescent cells, it may contribute to their ability to evade immune surveillance and clearance. So all these factors relate to Pi1 being a key component as a molecular amplifier of cellular senescence. Just one more component, one more little story about the Pi1, or a couple more things. This is work we just published in the JCI in December. So a lot of people challenged us when we originally made our observations about Pi1 the Amish. Well, and rightfully so. The contribution of confounding genes is an issue in a human population where a lot of people are related. And although I think we excluded that by doing additional studies and looking at doing GWAS on the carriers and controls, I think we were pretty well satisfied that Pi1 was the key driver of the aging related protection in the Amish. But we've done other things. We actually made a mouse. We engineered a mouse using CRISPR-Cas9 to perfectly replicate the genetic variant in the Amish population. So we call these TA700 mice. And so a heterozygous mouse that has that genetic phenotype has half the normal level of Pi1 in its blood. Homozygous animals for the TA700 variant have no Pi1 in their blood. So we have done a lot of experiments looking at how this impacts upon the mice. First, on the top of this slide here, this is additional data that we've accumulated in human beings over the last few years. We've studied things like their pulse wave velocity. We can see that carriers of the genetic variant have a lower pulse wave velocity. That is, their aorta is less stiff than unaffected members of the community. This is a preservation of vascular age or prevention of vascular aging in human beings. And if you look below, we see the same thing in aged mice that reducing Pi1 in animals protects them from vascular aging in a very powerful way. And interestingly enough, when we take Pi1, just one copy of Pi1 out of mouse, we get an extension of their lifespan that's almost 20%. It's about as big as anybody's reported in the last year. I mean, there was a lot of hoopla last year about IL-11 and impacting upon Mirroring lifespan, well, this is exactly the same magnitude of an effect, but actually it's already proven in human beings that it's protective in the natural experiment that Mother Nature's been doing. Now, we're not the only people that have found that Pi1 is related to, or maybe contributes to, or correlates with aging related morbidity. This is one component of the Grim age algorithm that Steve Horvath published about a few years ago. And then, probably the gold standard for measuring biological age in 2026. One of the key components of Grim age is a methylated DNA estimator of your plasma Pi1 level. So you don't have to measure Pi1 directly. You can just look at DNA and Pi1. But overall, DNA and Pi1 is the best predictor, standalone, of aging-related morbidity. Predicts overall mortality, predicts time to coronary disease, predicts a comorbidity index, predicts all kinds of age-adjusted morbidities like high blood pressure, type 2 diabetes, time to heart failure, age of menopause, visceral adiposity, fatty liver. When this paper came out, I called up Horvath and I said, I can't believe what you found here. It's like the other side of the coin. We would have predicted all this. We predict all this from the biology and the work that we've done in human beings. He said, I'm a statistical geneticist. I'm a mathematician. Pi1's not something I really know much about, but certainly it is a powerful predictor of these aging related phenomena. So I think this adds further evidence that Pi1 is a key driver to aging and aging-related morbidity in humans. I won't, it's too early in the day to talk about evolution, but suffice it to say, I think that Mother Nature is doing a really interesting experiment in Adams County, Indiana. I think it meets all the criteria for an evolutionary advantage for the reasons I've listed for you here. It's going to take some time, maybe a few million years for this to be borne out. But uh, Mother Nature has a subtle way of altering the fitness of our species, and it's not very common where you see a community that's culturally and geographically isolated to allow the potential for evolution to take place. So that's the story that got me into the field of aging. I'm not somebody that got here by writing a book and declaring myself an expert in the field of aging. We have plenty of people like that in this space. I've been working on a project and a program and questions for almost 40 years now, and this is where we are today. And now I'm very fortunate to have the opportunity to lead a new longevity institute that we have here at Northwestern. It's composed of multiple centers. For example, our Claude Pepper Center is a key component under the umbrella of our Longevity Institute, and we have several other centers in the Institute: nanoscience of aging, population science of aging, HIV and aging is a particular interest of ours. And John Potocsnak gave us the money to create this institute to actually do something about aging and people that are disadvantaged for one reason or another. In particular, an example is people with chronic HIV infections, but we extend that to other people with chronic conditions or circumstances that make them age more rapidly. I think this alone distinguishes our institute from almost any other program in the world. We're concerned about people that are aging too rapidly, and we want to normalize their rate of aging. We're not here to help billionaires live to be 150. One of the exciting things that we have is our human longevity laboratory. And I would just want to focus on that in the last few minutes I have with you today. So, if you were going to get your biological age measured recently, you'd have to go to somebody like this. It had some, they probably were occasionally right in predicting what your biological age and what your lifespan what might be, but the science wasn't very rigorous, and I don't think it was very reproducible. We've come up with a different way of measuring biological age, and a lot of this we learned by the measurements and the experience we've had in Adams County, Indiana. We've brought our learnings to Chicago, and with the help of Northwestern Medicine, they've invested in us and actually built out a dedicated space that I call the Human Longevity Laboratory. So if you come into our laboratory, we have a circuit that you go through. It takes about 2.5 hours to go through the whole circuit. And we have a number of stations that you stop at where we measure functional and physiological measures that are oftentimes known to change as a function of age. We have a neurosensory room. We have a cardiovascular room. That's my favorite room. And I'll spend a moment on that in a minute, but I like them all. We have a pulmonary room. We have a musculoskeletal room. We have a small gym where we can do all kinds of measures of strength and functionality and flexibility and stamina. And on top of the functional and physiological measures, we layer on deep molecular profiling, including things like epigenetic age. We just bought the instrument and it's being activated now on our campus to do broad platform proteomics using the Olink platform. We also do a measurement here on campus called glycan age, which is another index of biological age. We also do a series of measures that use AI-based tools that give us a readout of your biological age, including photograph of your retina, measurements of your gait and your mobility, your ECG age, and then we take all that information together, and then we give people this kind of information about what plot they fall on. Now, interesting, all of the estimates, all the measures of biological age don't always line up. We see differences from one to another. And this is an important point. I know epigenetic age is the gold standard for 2026, but I don't think it's perfect. And I think the best approach is to take kind of an agnostic approach and measure as much as you can, because we're still learning about this, and that's why we take an open-minded view about what's involved, and as we try to determine biological age in the patients that go through it. We've run almost 500 people through our laboratory now in the last 2 years, and we bring 2 people through a day in our laboratory. This is a fun slide. This shows the AI-based tools that we use in the longevity laboratory. We do a simple 12-lead electrocardiogram. I'm a cardiologist. I can read an ECG. I can't tell your age. We work with a group in Brazil that have analyzed millions of electrocardiograms, and they trained it to learn age. And we can do a 12 lead, pop it up in the cloud, and with our friends in Brazil, we can get back your biological age, your cardiac age in a matter of seconds. And if your age gap is significant, greater than 7 years, it predicts events in a relatively short period of time. We take a simple picture of your retina. We have you sit down and look at that little camera that you see right there. With a group in New Zealand called Toku who've analyzed hundreds of thousands of human retinal images, we can do the same thing. Pop the images up in the cloud. A few seconds later, we get your biological age back. And this tool even predicts things like kidney disease. With a group in Oxford, England, we do a simple 4-chamber echo. They can analyze it for age-related changes in the heart. It's actually diastolic dysfunction. And my colleague and collaborator here, James Cotton at Shirley Ryan, is a brilliant physician scientist. He's developed an AI-based tool using an iPhone where we can simply take a video of someone standing up, moving, walking, turning, and he can generate all kinds of critical information about gait speed, functionality, get up and go time, all that with a video that takes about 10 seconds to collect. So I want to spend a couple minutes talking about the key components of our measurements related to molecular profiling and biological age. I understand that epigenetics drive one of the versions of the biological clock. There are all kinds of things that impact upon the methylation of our DNA over our lifetime, but that pattern of methylation impacts upon the functionality of our DNA. And there are all kinds of things that can drive a maladaptive pattern of methylation across our genome. Everything from chronic stress to a poor diet to cigarette smoking to HIV infections. Fortunately, the epigenetic age is malleable. Although we know a lot of things accelerate it, we already are beginning to have some prospective evidence that interventions can actually impact upon biological age. Things like a healthy diet, losing weight, exercise, including aerobic exercise, some limited evidence that improving sleep patterns impacts upon your biological age. But this makes me optimistic. That makes me think that actually, if we can do something about biological age, if we can do something about epigenetic age, maybe someday we'll be able to show that plays out by slowing or reducing or the onset of age-relating illness and extending the health span. I told you we just got the Olink apparatus here on campus. This is amazing technology. I'm sure some of you already have it or know about it, but comes out of a company in Finland. I don't have stock in Olink. I wish I did. But in 2 microliters of plasma, you can get measures over 5,000 proteins for a price that's actually manageable. And if it's almost the Theranos idea, but this is not fraudulent. This is real. And people like Tony Weiss-Corey and his group have used Olink out at Stanford, and in that signature, in your proteins, in that panel of proteins that are on that plate, you can see your systemic age and the age of all your organs. So I think this is breakthrough technology. I think likely to certainly, it's certainly going to supplement and add to what we learn with epigenetic age. And I'm very excited to have this as part of what we offer in our aging program. So I'm not the only person involved here. We've got a huge team here at Northwestern involved in our human longevity laboratory, people from almost every discipline in medicine, including some world-class scientists, some potential future Nobel Prize winners. I'm hovering the arrow over their faces. These are world-class investigators that are involved with our program in a variety of different ways. We've got specialists from the pulmonary disease, in the musculoskeletal area, like James Cotton that I mentioned. Reproductive system aging is a big part of what we're doing, and Francesca Duncan and is one of our key members in that area. Josh is our AI guru in the aging space. John is the medical director or the research director of our program in the longevity lab, and Manjo Gill from ophthalmology is the key leader of our measures to measure age looking at the eye. So what we want to do, what we hope to do is to replicate our laboratory around the globe. We can't do everything here in Chicago. And maybe some of you that I'm talking to today might think about the idea that we create a domestic network around measuring biological age and testing interventions. What we propose is a standardized platform duplicated in laboratories around the globe where we collectively measure interventions in specific populations to see if we can find ways to slow down the curve of aging. Now, we already have some sites around the globe. We have a collaborator at Mount Sinai in Miami who'll be setting up his laboratory later this summer. We're far along in conversations with our friends in Brazil, as well as the dean of the medical school in Montevideo, to have a place in South America or two in the not too distant future. We just secured the funds to set up a laboratory in Stellenbosch, South Africa, and Northwestern has a long-standing partnership with that university. Northwestern has a partnership with the London Clinic and the Center of London, and we'll be putting a longevity laboratory there later this year. I have a long-term collaborator in the Pi 1 field at Tohoku University in northeastern Japan, and they're up and running already, and I'm actually co-director of the laboratory over there, but we need some other sites around the globe. We're talking to people in the Mideast. We definitely need to have something in India. We need to have another couple places in Europe. And I would hope that we could get some other places in the United States and go to the National Institute of Aging and get them to support a network that tests interventions that might bend the curve of aging, like the intervention testing network that's been active in mice for the last 30 years. I think it's time to do this in humans. We already have some research collaborations going on. We're already in the midst of a prospective study looking at the impact of stress reduction in human beings on biological age. We're doing a study with Tara Henderson, who's the chair of pediatrics here at Northwestern at Lurie. And we're looking at biological age in young adults that survived neuroblastoma. And we all know that surviving cancer and the challenges that provides with regard to chemotherapy and radiation likely impact upon biological aging, and we're doing this as a pilot so we can start testing interventions that might slow down aging. We have some other interesting uh, trials that will start later this year that involve dietary supplements. We have some other interesting things going on in the laboratory around looking at long COVID as a driver of accelerated aging, developing new wearable devices that measure something or factors that correlate with skin aging, and hopefully we're going to have a Pi1 inhibitor program relatively soon. My partner Toshio Miyata in Japan has developed an inhibitor that blocks Pi1 in human beings. It's already in being tested in Japan, and here I'm holding up a blister pack of drugs, a small molecule that inhibits Pi1, and we have a request to the FDA now to be able to begin testing this in human beings in Chicago relatively soon. So here we are. You know what? Everybody's been looking for the fountain of youth for a long time. I don't know exactly what it is. Maybe you do. Maybe you got a better idea than I do. But I do know that we have the tools and the resources and the expertise and the precision to be able to measure biological age in people and maybe do something about it. And the important thing is really, and I think many of you will agree with me, it's not how long we live. It's what's really important is how long we remain young. I got to thank key benefactors and partners and all this. Let me point out John Potocsnak and his family. So grateful to them. Perry McKay has given us real dollars and support for operating the longevity lab. Mary Smart and her foundation have also done that too. We've had money from the Impetus Award that helped us with in the work with the Amish population. I have on R35 that helps support our work around Pi1 and aging. And the endowment I have from Northwestern also helps with this. So thanks for your attention. I appreciate the opportunity to talk to you today.
Thank you so much, Dr. Vaughan. That was a fabulous talk. Really wonderful, really exciting. Let's see if we have any questions in the chat. And happy birthday, Dr. Vaughan, too.
It's relentless. Seems like I just had one. There it was.
Does anyone have any questions? You left them speechless. Well, we really, really appreciate you taking the time to speak with us today. This was really fascinating. And it's recorded, so I'm sure there'll be many people watching it at a later time as well. So we really appreciate it. It's a fabulous talk.
Well, very glad to do it. And I hope I can hear from some of you in other places around the country about the idea of creating a network that has kind of a standardized protocol and has a steering committee that comes up with ideas about patient populations we want to study and interventions that might be tested. There are lots of things out there that are on the radar screen that might have anti-aging effects, right? I mean, we all hear about them. Certainly the ones that have been talked about the most over the last few years are things like rapamycin and metformin, and I think it'd be worthwhile to put those agents through a rigorous protocol and actually prospectively see if they do anything beneficial. I'm open-minded to that. More recently, drugs like GLP-1 agonists have been suggested to have anti-aging properties. I don't think there's any real data on that yet in any meaningful way, but we all know that the best way, the most consistent way to lower biological age and extend health span or lifespan in animals, all kinds of animal species, is caloric restriction. And GLP-1 agonists will reduce your caloric intake, no doubt about it. There are other ideas out there. I told you we're testing a stress reduction program now. Um, there are other lifestyle interventions that might have an impact on biological age. There are other drugs that have been suggested that might have an impact, but the reality is we have the tools now to be able to measure this prospectively in a relatively short period of time. And what we want to hope we can do over time is be kind of the kind of bring some adult supervision to the room of aging. It's kind of crazy out there. A lot of weird things are being suggested as being helpful, but I don't think we have to study thousands of people for years to get a readout that something might have the potential. We can do proof of concept studies in, I think, 3 to 6 months and look at the readout and then move on to the next one and then see, try to bring some rigor and some science to this field that has been lacking in some ways so far.
That's a great point. And then you did get one question in the chat. Are there limitations to any of the tools based on patient pathology? Specifically, was thinking of the retinal scan in folks with diabetes as an example.
Yeah, that's a great question. When you develop an AI-based tool and you do machine learning, you don't exactly know what the machine is learning on. Its patterns, it doesn't necessarily tell you. It's clearly mostly about the microvasculature and factors like hypertension and diabetes do accelerate biological age using this tool. It's reading something about changes in the microvasculature, and that's probably no surprise.
Right. Well, if there's no further questions, again,
Okay.
thank you for Dr. Vaughn for a great talk, and thank you for everyone for joining us today. We hope to see you again next month for our April Pepper Center rounds. Thanks, everyone.
Thank you. Thanks, Sarah.
Thank you.